When we picture the animal kingdom, we almost exclusively imagine animals with bones: mammals, birds, reptiles, fish. But this focus represents a massive statistical anomaly. Look at this diagram mapping animal diversity. Vertebrates make up barely 3% of all animal species on Earth. The remaining 97% forms a vast, dense web of invertebrate phyla. What unites this overwhelming majority is a structural absence. They completely lack a cranium and a defined vertebral column. It is a mistake to view these organisms as primitive. They are the original evolutionary laboratory. Long before bone existed, these animals engineered every essential piece of biological hardware we rely on today. To truly understand human biology, we have to deconstruct the biological machines that first invented cuticles, venom glands, and centralized nervous systems. The extreme physical adaptations developed by these organisms now serve as the foundation for modern pharmacology and medicine. Take the predatory conus snail. The neurotoxins it uses to paralyze prey are being adapted into targeted pain relief. This 3D molecular animation shows a conotoxin peptide physically blocking a neural ion channel, halting the electrical impulse of a pain signal. The medical leech, Hirudo medicinalis, manufactures a proprietary anticoagulant called hirudin. This compound is isolated and applied in clinical settings to treat severe human circulatory disorders. Or consider the nematode Caenorhabditis elegans. Its predictable cellular development makes it the ultimate model organism for mapping genetic pathways in the lab. Harvesting these biomedical tools requires us to reverse engineer the phylogenetic tree. We need to look at how and why these specific physiological traits evolved to guarantee survival in their original environments. The baseline for multicellular animal life begins with the phylum Porifera, the sponges. They're classified as parazoa, meaning they operate entirely with true embryonic tissues. Water is drawn through small ostia, pools in the central spongocoel, and is expelled through the osculum. This flow is driven by choanocytes. Whipping their flagellum, they generate the current to phagocytose passing bacteria. The structural similarities here point directly to common ancestry. Notice the exact resemblance between the free-living choanoflagellate at the top and the embedded choanocyte cell below it. When environments become hostile, sponges utilize an extreme survival tactic, packing essential cells into protective capsules called gemmules to endure until conditions improve. The first major architectural upgrade appears in the phylum Cnidaria. Here, animals develop true tissues, specifically a diploblastic structure with an ectoderm and endoderm, arranged in radial symmetry. Cnidarians also engineered a highly specialized biological weapon. This is the cnidaria. When the external hair trigger is touched, it everts a coiled pneumatocyst at 40,000 Gs, instantly injecting a paralyzing neurotoxin into its target. This phylum also includes the cubozoa, or box jellyfish, which possess image-forming eyes complete with a cornea, lens, and retina. They process visual data and hunt actively, entirely without a centralized grain. These basal animals establish the rules for cellular cooperation and offensive hardware. However, their radial design severely restricted their capacity for directed, efficient mobility. The superphylum Lophotrochozoa solved this mobility problem by adding a third embryonic tissue layer, the mesoderm. As this abstract model shows, the addition of the mesoderm allows the animal blueprint to transition from a radial circle into a bilaterally symmetrical shape. Bilateral symmetry requires cephalization. To navigate forward motion, the organism must concentrate its nervous tissue and sensory organs at the anterior or front end of the body. The phylum Platyhelminthes, the flatworms, utilizes this bilateral blueprint. They are acoelomates, meaning their solid bodies lack an internal cavity. They must remain flat so gases and nutrients can diffuse directly across their tissues. Within this group, the parasitic tapeworms, or Cestoda, demonstrate the evolutionary logic of regression. A tapeworm completely abandons the newly evolved digestive system. It absorbs nutrients directly from the host's gut through its outer surface, the tegument. This allows the animal to allocate maximum energy exclusively to reproduction. Biological evolution does not strictly favor greater complexity. In specific environments, the most efficient survival strategy is to strip hardware back to its bare minimum. The phylum Mollusca pursued a different architectural strategy. standardizing a highly successful three-part blueprint: a muscular foot for locomotion, a visceral mass containing internal organs, and a mantle to secrete a protective shell. During embryonic torsion, the snail's visceral mass rotates 180 degrees, bringing the anus directly over the head. Despite the sanitation risk, it allows the sensitive head to retract into the shell first, blocked by the tough foot. Cephalopods, like squid and octopuses, modified this exact same three-part blueprint into a closed circulatory system with image-forming eyes, engineering a high-speed, highly intelligent predator. Moving to the phylum Annelida introduces the next major structural breakthrough: metamerism, or true body segmentation. By dividing the coelomic cavity into repeating compartments separated by physical walls, annelids can generate localized hydraulic pressure. This allows for independent muscle control in different parts of the body simultaneously. Dividing a body into isolated, repeating mechanical units was the exact engineering leap required to achieve advanced, energy-efficient locomotion. The superphylum Ecdysozoa approaches survival through defensive engineering. Their unifying characteristic is a rigid, protective outer cuticle. The physical cost of this armor is that the organism cannot grow continuously. To expand, it must undergo ecdysis, periodically shedding its outer layer in a vulnerable molting process. Within this group is the phylum Nematoda, which includes the roundworm Chinorhabditis elegans, an organism whose cellular mapping makes it invaluable to researchers. C. elegans exhibits eutyly. It reaches a genetically fixed, exact number of somatic cells. It develops exactly 959 cells, providing scientists with a pristine, perfectly mapped biological canvas. Contrast this laboratory model with pathogenic nematodes, such as Wuchereria bancrofti, which deploy this same tough anatomy inside human hosts. These parasitic worms are responsible for elephantiasis. Their physical accumulation directly obstructs the human lymphatic vessels, preventing fluid drainage and causing severe, localized edema. The ecdysozoan architecture requires strict limitations. Their rigid external constraints yield unmatched defense, while their rigid internal cellular maps make them perfect subjects for decoding human genetics. The microscopic phylum Tardigrada, the water bears, act as the ultimate stress testers for this ecdysozoan body plan. To survive hostile environments, they enter cryptobiosis. This is an extreme state of suspended animation triggered by desiccation, freezing, or high radiation. During cryptobiosis, water molecules evacuate from the cells and are replaced by specialized protective proteins. This geometric matrix stabilizes the cell membrane, preventing any tissue destruction until moisture returns. The molting cuticle strategy reaches its absolute apex in the phylum Arthropoda. Here, rigid chitinous exoskeletons are combined with jointed appendages for specialized movement. Arthropods currently represent roughly 85% of all known animal species. Statistically, they are the most mechanically successful design in Earth's history. The armored, segmented architecture of the ecdysoans proved to be the winning biological formula, allowing them to dominate almost every terrestrial and aquatic biome on the planet. Looking at this complete phylogenetic tree, we can trace the precise trajectory of biological engineering. Life transitioned from the tissueless fluid dynamics of early sponges to the precise cellular mapping of nematodes to the indestructible armor of tardigrades. These are not primitive animals. They are highly specialized, mechanically optimized survival machines. The stabilizing proteins that allow tardigrades to survive cryptobiosis are now being studied to stabilize vaccines, allowing for global transport without refrigeration. The neural complexity of cephalopods and the high-speed neurotoxins of cnidarians are guiding the future of neurology and pharmacological pain management, and the rigid genetic map of the nematode remains critical to understanding human cellular disease and programmed cell death. To protect natural ecosystems and push the boundaries of human medicine, science has to look past the 3% of vertebrates that resemble us. We have to fully embrace the mechanical genius of the spineless majority.